Chemogenetics is an innovative technology where new, unnatural receptors are created that exclusively bind drug or other small molecules that can be administered by experimenters. We will be focusing on the most popular chemogenetic strategy used in neuroscience, called designer receptors exclusively activated by designer drugs, otherwise known as DREDs. DREDs aim to change the electrical properties of a cell when they bind to their ligand, providing a precise, non-invasive method to activate or inhibit neurons and glial cells in freely moving animals. By selectively expressing these receptors in our cell population of choice, we can administer the small molecule throughout the body and still see specific effects on our target neural cells. This cell-type specific control provides the platform to investigate the relationships between neurons, glial cells, and behavior for both translational and basic applications. DREDs are modified G-protein coupled receptors, meaning they function through secondary messenger cascades. DREDs have been designed for a variety of GPCR types. Some of the most common DREDs are mutated muscarinic acetylcholine receptors. These mutant receptors no longer bind to acetylcholine and instead only bind to a small drug called clozapine N-oxide, abbreviated CNO. CNO can be given orally, for example, in drinking water or food. When CNO activates the DRED, the associated G protein will initiate a signaling cascade. We can express different kinds of G proteins to control what effect that signaling cascade has. DREDs that use the G protein called GQ lead to the opening of calcium channels, which allows calcium to flow into the cell. If this cell is a neuron, this depolarization will increase its chance of firing an action potential. Another type of G protein, the GI protein, has the opposite effect. It leads to hyperpolarization of a neuron, making it less likely to fire. One way that this can happen is through the opening of potassium channels, allowing potassium to flow out of the cell. We can control which cell populations express the DRED through a combination of genetic strategies and location-specific delivery of the DRED transcript. Now we will walk through the process of conducting a chemogenetic experiment. There are many factors to consider when planning a chemogenetic experiment. First, what is the cell population that you are trying to target? Chemogenetics is a very flexible technique and can be adapted to target many subtypes of both neurons and glial cells. We can often target specific cell types by designing our DRED transgene to be under the control of a promoter that is only active in specific cell types. We call this a cell type specific promoter. Second, which brain region do you want to target? We often use viruses to control where the DRED genes are being delivered. Viruses naturally want to infect cells with whatever DNA they are carrying. By infusing viruses carrying our DRED genes in a specific brain region, we can control where in the brain the DRED might be expressed. Lastly, how do you want to manipulate your chosen target population? As mentioned, there are many different types of modified receptors, and each will manipulate your target cell population in a different way. For example, using a muscarinic GQ DRED will activate your target cell, while a muscarinic GI DRED will inhibit it. Once you have decided your target cell type, location, and manipulation, you are ready to start your chemogenetic experiment. In the example we will show here, we use virally mediated gene transduction to achieve specificity in receptor expression. As we discussed earlier, this technique utilizes the infectious property of viruses to introduce the receptor gene into cells under the control of a cell type specific promoter. In this experiment, we are using a GQ DRED with a promoter that will drive expression in adult-born neurons. Our cell population of interest is in the subgranular zone of the hippocampus, so that is where we will be targeting our viral delivery. Following viral delivery, we waited 2 weeks for the drug to be expressed on our target cells before moving on to administration of the agonist. Now that the receptor is expressed in our targeted cell population, we can begin to manipulate these cells with administration of the receptor agonist. In this experiment, we are adding CNO to the mouse's drinking water so that we can study the effects of long-term activation. Remember, when CNO is administered, it binds only to the DREAD expressed in the adult-born neurons in the hippocampus, causing them to fire more action potentials through the GQ protein mechanism. DREAD activation can be done in awake, freely moving animals, allowing us to see how changes in cell firing can change the animal behavior. In this example, we are looking at a zero maze, which measures an animal's anxiety-like behaviors. This maze is a circle split into quarters, with open and closed sections. Mice are prey animals, meaning that in high-stress situations, they tend to hide in the dark. Therefore, we use their time in the closed arm versus the open arm of the zero maze as a measure of anxiety-like behavior. By using DREAD manipulations, we are able to see profound effects on this type of behavior in mice. As you can see, the mouse that had CNO is displaying less anxiety-like behavior by exploring both the open and closed parts of the zero maze, compared to the mouse without CNO that is hiding in the closed portion. This result suggests that after a series of prior manipulations, the last step of activating the adult-born neurons in the hippocampus causes reduced anxiety-like behavior. Chemogenetics provides the platform for specific, non-invasive manipulations of targeted cell populations. This allows scientists to investigate neural circuits and the relationships between these circuits and behavior. Despite its many uses, this technique is limited in the time it takes for systemic administration of CNO to activate your target cells. If you're looking for tight temporal control over your experiment, you may want to use a technique like optogenetics, which allows you to turn on and off activation within seconds. Along with other powerful neuroscience tools, chemogenetics contributes to the expansion of the neuroscience field, both in the understanding of the brain and behavior, as well as in treatment development for neurological and neuropsychiatric diseases.